Hey there, curious minds, and welcome back to the Deep Dive. Today, we're pulling back the curtain on some truly mind-bending chemistry that's happening inside every living cell, every single second. We're diving into the fascinating world of biological inorganic chemistry, drawing our insights from Chapter 27 of Shriver and Atkins Inorganic Chemistry, 5th edition.
Indeed. Our mission is to explore how life, from the simplest bacteria right up to complex mammals, has ingeniously harnessed elements beyond just the usual suspects: carbon, hydrogen, oxygen, and nitrogen. We're talking about crucial metal ions, iron, zinc, copper, even things like molybdenum and how they are absolutely essential for everything, really. Sparking reactions, transmitting signals.
Building structure, sensing what's going on around them.
Exactly. You're going to gain, hopefully, a unique understanding of these vital roles, seeing how these concepts all fit together, how they intertwine to create the very fabric of life.
Get ready for some surprising facts and maybe a few aha moments that will completely change how you view your own biology. Let's jump in.
Okay, so our journey begins with the real fundamentals, living cells. Whether they're simple prokaryotes like bacteria or the more complex eukaryotes you find in animals and plants, they all share this crucial feature: membranes.
Right. These incredibly thin layers, just lipid bilayers, about 4 nanometers thick, they act as really sophisticated barriers.
But they're not just passive walls, are they?
Oh, definitely not. They're actively managed. They contain these special protein molecules that form channels and pumps. And this controlled movement of ions allows for something we call compartmentalization.
Compartmentalization,
Mhm.
like keeping things in separate boxes.
Precisely. The precise segregation of specific elements inside and outside the cell, and even within internal subcompartments, organelles like the nucleus for DNA or mitochondria as the cell's tiny fuel cells.
Hmm.
This whole dynamic balance, keeping the right ion levels in the right places, that's homeostasis. It's amazing when you see this balance in action. Take potassium, K plus A, and sodium, Na plus A. There's this stark contrast across the cell membrane, right?
Huge contrast. High K plus inside the cell, but much lower Na plus inside.
And that's not just how it is, the cell makes it that way.
Absolutely. It's actively maintained, and this gradient creates an electrical potential difference across the membrane. It's like a tiny battery, essentially, storing energy for the cell.
Wow. And this precise control, it applies to other ions too.
Oh, yes. Calcium, K2 plus A, is a great example. It's kept at incredibly low free concentrations in the main part of the cell, the cytoplasm. We're talking less than 1/10,000th of a molar concentration. But then it's actively pumped into specific organelles. This makes it super sensitive for signaling. Even a tiny increase in free calcium can trigger a huge cellular response.
So beyond the big four, carbon, hydrogen, oxygen, nitrogen, life uses this really diverse array of elements. Phosphorus and sulfur are major players too, of course.
And sodium, magnesium, calcium, potassium, also pretty abundant. But then you get the critical trace elements.
Right, including lots of D block metals, iron, zinc, copper, molybdenum.
And others like selenium, iodine, even silicon and boron in some cases.
So how did nature choose these particular elements?
Well, it often comes down to a combination of availability, what was actually around when life evolved, and useful inherent properties.
Mhm.
Zinc, for instance, is widely used partly because it's quite abundant and generally non-toxic. Copper, on the other hand, was pretty scarce on early Earth, trapped as insoluble sulfides. It only really became a major player after oxygen appeared in the atmosphere.
Okay, so these metals are crucial, but where do they actually connect with biology?
Yeah.
They aren't just floating free, right?
No, absolutely not. That would often be toxic. They interact strongly and very specifically with biological molecules, mostly proteins, but also nucleic acids like DNA. DNA's structure is actually stabilized by weak interactions with ions like potassium and magnesium coordinating to its phosphate groups.
Interesting. But the main action is with proteins.
Exactly. We spend a lot of time talking about metalloproteins. That's proteins containing one or more metal ions. They are absolutely central to biological inorganic chemistry. These metals allow proteins to do amazing things. They help with oxidation reduction reactions, drive radical-based rearrangements, catalyze the breaking of bonds with water hydrolysis, and play key structural roles too.
Like the calcium example, binding changes the protein's shape.
Precisely. When K2 plus binds, it often causes a change in the protein's conformation, its 3D shape. And that change is often the key step that kicks off a whole cell signaling pathway.
And the proteins themselves are kind of designed to grab these metals, aren't they? With specific donor groups.
Yes, exactly. These come from the amino acid side chains within the protein structure. You have hard donors, like the oxygen atoms in the carboxylate groups of aspartate or glutamate. They tend to prefer ions like calcium. Then there's the nitrogen atom in the imidazole ring of histidine that's incredibly important for binding iron, copper, and zinc.
And sulfur, too.
Right, the soft thiol sulfur from cysteine. It's great for binding metals like iron, copper, and zinc, but it's also why toxic heavy metals like cadmium and mercury can be problematic. They bind strongly to cysteine, too.
And besides the standard amino acids,
Nature also uses some special ligands. These are large organic macrocycles, big ring structures. The most famous is probably the porphyrin ring, like the one holding iron in hemoglobin or magnesium in chlorophyll. What's really fascinating, though, is how the protein structure itself can sometimes force the metal into an unusual coordination geometry.
what do you mean by unusual?
Well, a shape it wouldn't normally adopt if it were just in solution. The protein can kind of twist the metal's surroundings, maybe into a shape that resembles the transition state of the reaction it needs to catalyze.
Wow, so it's biasing the reaction, making it easier.
Exactly. It's an incredibly elegant way nature speeds things up and controls chemistry.
Okay, let's shift gears a bit. Talk about transport, signaling, and energy flow. Metals moving around. We mentioned sodium and potassium earlier, the gatekeepers.
Right. And we need to distinguish how they move. There's passive transport ions flowing down their concentration gradient, usually through ion channels.
Yeah.
And then there's active transport moving ions against their gradient, which requires energy usually from breaking down ATP.
Yeah.
This is done by ion pumps.
And the K plus channel you mentioned is an example of passive transport.
Yes, and it's just a marvel of selectivity. Think of the structure. A central watery cavity that leads into a narrow part called the selectivity filter. This filter is lined with precisely spaced oxygen atoms from the protein backbone. Potassium ions, K+, fit perfectly here. They shed their surrounding water molecules and interact with these oxygens. They queue up and sort of nudge each other through. It's called a concerted displacement mechanism.
Okay, but how does it keep sodium out? Sodium ions, Ni plus say, are smaller. Shouldn't they fit?
That's the brilliant part. It's not about being too small. The cavity is actually too large for Na+ to interact effectively with all those oxygen atoms simultaneously.
Yeah.
It can't shed its water coat as favorably, so the channel doesn't bind K+ super tightly. That would slow things down. Instead, it relies on this fast, weak binding that perfectly discriminates. The result, an amazing 10,000-fold selectivity for K+ over Na plus air.
10,000 to 1. That's incredible molecular engineering.
It really is.
And working the other way, you have the Na plus K plus pump, the active transporter.
Exactly. The Na plus K plus NACE ATPase. It actively pumps 3 sodium ions out of the cell and 2 potassium ions in for every molecule of ATP it breaks down. This process involves the protein cycling through different conformational changes, different shapes, powered by that ATP hydrolysis. It's absolutely vital for maintaining cell volume, nerve impulses, all sorts of things.
Okay, so that's sodium and potassium. What about calcium, the messenger?
K2+, yeah. It's the rapid intracellular messenger. Especially in higher organisms, its unique properties make it perfect for this.
Like what?
Well, it has fast ligand exchange rates, meaning it can bind to things and let go really quickly. It has intermediate binding constants, not too strong, not too weak, and a large, flexible coordination sphere. It's not too fussy about the geometry of what it binds to.
So it can react fast and trigger things quickly?
Precisely. Take a protein called calmodulin. It's a small regulatory protein with 4 sites that can bind K2 plus chiromis. When calcium levels rise slightly in the cell, K2 plus ions bind to calmodulin. This binding causes a huge conformational change in calmodulin. It literally changes shape. And this new shape allows it to interact with and activate various target enzymes like protein kinases, kicking off a whole cascade of cellular responses.
And the speed is key. You mentioned it's much faster than magnesium.
Way faster. Calcium's ligand exchange is something like 1,000 to 10,000 times faster than magnesium's. That's essential for processes that need to happen quickly, like muscle contractions or neurotransmitter release.
All right, let's turn to zinc now. You said it's more structural and regulatory.
That's its main role, yes. Zinc 2, or Zn2 plus for Zem, prefers to form stable complexes, often with donors like the nitrogen and histidine or the sulfur and cysteine residues within proteins.
And the classic example is zinc fingers.
Exactly. These are distinct protein folds, little structural motifs, where a zinc ion holds the protein chain together in a specific shape. Often it's coordinated by 2 cysteines and 2 histidines. This stable structure, the finger, is then perfectly shaped to recognize and bind to specific sequences of DNA-base pairs. This is crucial for controlling gene transcription, turning genes on or off.
And importantly, zinc itself doesn't usually change its oxidation state in these roles.
Right. Zin2+ is redox inactive under biological conditions. It doesn't easily gain or lose electrons. That's vital when it's interacting with something as sensitive as DNA. You don't want it triggering unwanted oxidative damage, which metals like iron or copper could potentially do.
Speaking of iron, you called it a paradox earlier. Essential but toxic and hard to get.
Yes, a major challenge for life is simply acquiring iron. In the oxygen-rich world we live in, iron mostly exists as iron, Fa3, and Fa3 forms very insoluble hydroxides at neutral pH, basically, rust. It's not available.
So how does life solve that?
Well, bacteria have a clever trick. They secrete small molecules called cytophores, things like enterobactin or ferrochrome. These are like specialized claws, highly specific for Fa3, with incredibly high binding affinities association constants up to 10 to the power of 52. They effectively scavenge any available iron from the environment.
Wow. And in higher organisms like us.
We use proteins. Transferrin is the protein that transports iron through the bloodstream. Interestingly, it needs a helper ion, bicarbonate, to bind iron tightly. Binding iron causes a big conformational change in transferrin, and for storage, inside cells, we use ferritin. This is an amazing protein. It's like a hollow sphere that can store up to 4,500 iron atoms inside its core, safely tucked away as a hydrated iron 3 oxide mineral.
So it stores it as Fa3, the less soluble form.
Yes. It has special sites called peroxidase centers that oxidize incoming Fa2 to Fa3 for storage. Then, when the cell needs iron, it reduces the Fa back to the more mobile Fa for release. Very elegant control.
Okay, let's move to oxygen, O2. Essential for us, but you mentioned it's tricky to handle. Low solubility.
Low solubility in water, yes, which means we need ways to transport it efficiently. And it can be toxic, too. That's where metalloproteins come in again, this time as O2 carriers.
Like myoglobin in muscles?
Right. Myoglobin, or MMU, stores oxygen in muscle tissue. It's an iron protein. In its deoxygenated form, the iron 2s is 5-coordinate and sits slightly above the flat porphyrin ring. The protein looks sort of bluish-red. But when an O2 molecule binds, the iron becomes 6-coordinate, moves down into the plane of the porphyrin ring, and the whole thing turns bright red, that familiar color of oxidated muscle or blood.
And hemoglobin in blood is similar.
Hemoglobin, HB, is the main O2 transporter in red blood cells. It's actually like 4 myoglobin units packed together, a tetramer. And this structure allows for something really important: cooperative binding.
Cooperative binding.
Yeah. It means that when the first O2 molecule binds to one of the 4 subunits, it causes a small structural change that makes it easier for the next O2 to bind to another subunit, and so on. This results in a sigmoidal O2 binding curve, S-shaped. It means hemoglobin is very efficient at picking up lots of oxygen where it's abundant, like in the lungs, but also good at releasing it where it's needed, in the tissues where O2 concentration is lower. That conformational change is the key.
Fascinating. Okay, underlying all this is energy. You mentioned electron flow.
Right. Life ultimately gets its energy, directly or indirectly, from the sun. And this energy is often harnessed through the controlled flow of electrons, usually from some kind of fuel molecule to an oxidant, like oxygen.
And in organisms, this happens in chains.
Yes, along electron transport chains. These chains involve specialized electron transfer centers, often metal-containing sites built into proteins. Common examples are FES clusters, cytochromes, and certain copper sites. These centers are optimized for fast and efficient electron transfer. A key property is having low reorganization energy.
Low reorganization energy. What does that mean practically?
It means the structure around the metal center doesn't have to change much when an electron is added or removed. Think of it like a well-oiled machine part. It moves smoothly without needing a big rearrangement each time. This makes the electron transfer fast. They also often facilitate electron tunneling electrons jumping quantum mechanically over short distances, usually less than about 1.4 nanometers.
You mentioned cytochromes.
Yes, cytochromes are often colored proteins, pigments containing an iron porphyrin group, similar to hemoglobin, but used for electron transfer, not O2 transport. They cycle between Phe3 and Phe states. Their coordination environment changes very little during this, making them excellent electron carriers.
And FES clusters.
Iron-sulfur clusters. These are really widespread and ancient. They typically involve iron atoms bridged by sulfide ions and also coordinated by cysteine sulfur atoms from the protein. You find different arrangements like 2Fe2s, 3Fe4s, and the common 4Fe4s cubane-like structure. They often handle electrons at more negative potentials than cytochromes, And finally, there are blue copper centers found in proteins like plastocyanin. They get their intense blue color from charge transfer transitions. They have high reduction potentials, and their protein environment is very rigid, again ensuring that minimal reorganization energy for really efficient electron transfer.
Amazing diversity. Okay, let's switch to catalysis, making reactions happen. How do metals help there, especially with acid-base reactions?
Well, in biological systems, you usually can't just dump in strong acid or basive. The pH needs to stay near neutral. Metal ions provide a much more controlled way to do Brensted acid-base catalysis.
And zinc is a key player here.
Zinc enzymes are fantastic examples. Zinc has this great combination. It's reasonably abundant, it's redox inactive, so it won't cause unwanted side reactions. It forms strong bonds to protein donors, but it also allows rapid exchange of other ligands, like water.
Okay, let's unpack this. A specific example.
Carbonic anhydrase, or Ca. It's one of the fastest enzymes known turnover rate, around 1 million reactions per second. Its job is to rapidly interconvert carbon dioxide, CO2, and bicarbonate, HCO3, crucial for getting CO2 out of tissues and regulating pH.
1 million per second. How does zinc help achieve that?
It uses a Zn hydroxide mechanism. The zinc ion is typically held by 3 histidine nitrogens, and the fourth coordination site binds a water molecule. The positive charge of the Zn2 ion makes that bound water much more acidic. It lowers its pKa significantly. So the water readily loses a proton, H+, forming a zinc-bound hydroxide ion. This hydroxide is a powerful nucleophile. It readily attacks the CO2 molecule. Then, a clever network of hydrogen bonds helps shuttle protons away quickly, regenerating the enzyme. That proton transfer is actually the rate-limiting step. It's so fast.
Incredible coordination. Are there other metal catalysts?
Oh, absolutely. Think about magnesium enzymes. Rubisco, for instance, is arguably the most abundant enzyme on Earth. It uses Mg2+ at its active site to fix atmospheric CO2 during photosynthesis, basically the start of making biomass.
And iron, too.
Yes, iron enzymes can be catalysts. Aconitase, which we mentioned in electron transport, is part of the citric acid cycle. It uses its 4Fe4S cluster catalytically to isomerize citrate to isovitrate, and one specific iron atom in that cluster actually binds directly to the substrate.
Okay, so metals help with basic chemistry. What about dealing with oxygen catalytically, reducing it or using it?
Life has evolved amazing systems for this too. Peroxidases, for example, deal with hydrogen peroxide, H2O2, which is a toxic byproduct of metabolism. Enzymes like horseradish peroxidase use an iron porphyrin center. They actually form isolable intermediates during their cycle. What was historically called compound I is actually in Fe-O species coupled with a radical on the porphyrin ring. This shows iron reaching these higher oxidation states in biology.
Then there are oxidases.
Right, oxidases reduce O2 directly, usually to water or peroxide, but without incorporating the oxygen atoms into another molecule. The king here is cytochrome Eth oxidase. This is a crucial enzyme in the mitochondrial membrane, the final step in the respiratory electron transport chain in complex life. It performs the tricky 4-electron reduction of O2 all the way to water, and importantly, it acts as an electrogenic proton pump. It uses the energy released from electron transfer to pump protons across the membrane, building up that gradient we use to make ATP. It's fundamental.
So oxidases just reduce O2. What about enzymes that put oxygen into molecules?
Those are oxygenases. They catalyze the insertion of one or both oxygen atoms from O2 into an organic substrate. Monooxygenases insert just one O atom, reducing the other to water. The most famous family is cytochrome P450. These are heme-containing enzymes found everywhere, involved in detoxifying compounds, making hormones, lots of things.
P450. How does it work? How does it insert that oxygen atom?
It uses a fascinating oxygen rebound mechanism. It activates O2 to form a highly reactive intermediate, a Fe-IVO ferrule species, similar to peroxidase compound ALR, but without the porphyrin radical. This ferrule species is powerful enough to abstract a hydrogen atom from the substrate molecule. Then, in a rebound step, it transfers the OH group back onto the substrate radical, effectively inserting an oxygen atom. It's like the enzyme tames a superreactive oxygen atom just long enough to do precise chemistry.
Wow. And what about the biggest oxygen reaction of all making it? Photosynthesis.
Ah, yes. The Oxygen Evolving Center, or OEC, in photosystem 2. This happens in plants, algae, cyanobacteria. It's truly one of nature's most incredible feats of chemistry. The OEC is this cluster containing 4 manganese atoms and 1 calcium atom, arranged with bridging oxygen atoms. Essentially a tiny piece of manganese oxide. Its job is to catalyze the 4-electron oxidation of 2 water molecules to produce 1 molecule of O2, releasing protons and electrons. This process, starting maybe 2.5 billion years ago, generated basically all the oxygen in our atmosphere.
4 manganese atoms and a calcium. How does it work?
Light energy drives the process. Successive photons hit photosystem 2, and each one allows the OEC to lose an electron, stepping through a cycle of oxidation states, usually labeled S0 through S4. When it reaches the highly oxidized S4 state, it can finally oxidize water and release O2, resetting back to S0. The manganese ions are key because they can access multiple stable oxidation states like +2, +3, +4, and the calcium ion seems crucial too, likely helping to bind the water molecules in the right place and stabilize the structure without getting oxidized itself.
Mind-blowing. Are there other catalytic metals we should touch on? Cobalt?
Cobalt is interesting. It's mainly used by microorganisms which make vitamin B12 and its derivatives. The cobalt sits in a corin ring, similar to porphyrin but slightly different. A key form is coenzyme B12, which features a unique direct cobalt-carbon bond to an adenosine group.
A metal-carbon bond in biology. That's rare.
Very rare and very important. Cobalt can cycle between Co3, Co2, and even Co states. The Co form is a supernucleophile used in methyl transfer reactions, like making the amino acid methionine. But coenzyme B12 is famous for driving radical-based rearrangements. The relatively weak Co-C bond can break homolytically, one electron going to cobalt, making stable Co, the other creating a reactive carbon radical. This radical then initiates complex rearrangements within the enzyme's active site, controlled radical chemistry.
And molybdenum, tungsten.
Right, the heavyweights. Molybdenum is widespread. Tungsten is found mainly in some prokaryotes. They often work with a special cofactor called molybdopterin. Their specialty is oxygen atom transfer reactions, but where the oxygen atom usually comes from water, not O2, enzymes like sulfite oxidase use molybdenum cycling between Mohs base and Mohs to add an oxygen atom from water to sulfite, making sulfate.
So catalysis is huge, but metals also act as sensors, detecting things.
Absolutely. Organisms need to sense their environment. Many metalloproteins act as sophisticated sensors for small molecules like O2, nitric oxide, NO, carbon monoxide, CO, or even the levels of essential metals like copper and zinc. Sensing these triggers adaptive responses.
How can an iron protein sense something?
Take those FES clusters again. They can be sensitive to oxygen or iron levels. If oxygen is high, a 4Fe4S cluster might degrade to a 3Fe4S cluster, or the iron might even be lost entirely. This change in the cluster alters the protein's overall conformation, its shape. That shape change can affect whether the protein binds to DNA or RNA, switching genes on or off. The iron regulatory protein, IRP, is a classic example. It senses cellular iron levels and controls the production of ferritin for storage and transferrin receptor for uptake.
So the metal cluster itself is the sensor. What about sensing gases like NO or CO?
Heme sensors are often involved there. For example, the enzyme guanylate cyclase has a heme group that binds NO. This binding displaces another ligand, causes a conformational change, and activates the enzyme to produce a signaling molecule. Bacteria have similar heme-based sensors for CO, like CUA.
And sensing metal levels themselves, copper and zinc.
Yes, cells need to control these very carefully. Too much copper or zinc is toxic. E. coli, for example, has a protein called CUR. It's a transcription factor that specifically binds copper. It uses two cysteine residues to bind E. coli in a linear geometry. This binding flips a switch, changing the protein shape so it can bind DNA and turn on genes for a copper export pump. Get rid of the excess copper. There's a similar sensor, ZenTR, for zinc 2, which binds it tetrahedrally using cysteine and histidine ligands, controlling zinc export. It's all about maintaining that strict metal homeostasis.
Incredible feedback loops. Okay, shifting gears again. What about building things, structures, biomineralization?
Right. Life doesn't just use metals for catalysis and signaling. It uses inorganic minerals to build amazing structures, things far more intricate than we can usually make synthetically.
Like shells and bones.
Exactly. Seashells, eggshells, those are often calcium carbonate, calcite or aragonite. Bones and teeth are primarily calcium phosphate in the form of hydroxyapatite. These are incredibly durable materials.
Any more exotic examples?
Oh yeah. Some bacteria, magnetotactic bacteria, make tiny perfect crystals of magnetite, which is iron oxide, F3O4. They arrange these crystals in a chain inside the cell.
Why?
It acts like a tiny compass needle. It allows the bacteria to navigate along the Earth's magnetic field lines, helping them find their preferred low-oxygen environment in sediments, microcompasses. Plants also use silicon dioxide to build tough, protective structures.
So it's not just dumping minerals down, it's control.
Highly controlled. It ranges from simple precipitation dictated by solubility, all the way up to intricate constructional control, where the organism precisely templates mineral growth using organic molecules like proteins. Bone is a fantastic example of this composite material, inorganic crystals interwoven with organic collagen fibers.
Amazing. Okay, one last big area, metals in medicine. Using these principles, or even metals not normally found in biology, for treatments.
Absolutely. Inorganic chemistry has made huge contributions to medicine. One area is chelation therapy.
Chelation, like grabbing onto metals.
Exactly. It's used to treat metal overload conditions. For example, in genetic disorders where people accumulate toxic levels of iron, doctors can administer ligands like desferrioxamine. This molecule was inspired by bacterial cytophores. It binds very tightly to the excess iron, forming a complex the body can excrete.
So you're removing harmful excess metal. What about adding metals for therapy?
The most famous example is probably cisplatin in cancer treatment, cis-BTCl2, NH3-2. Its discovery was actually serendipitous. It works primarily by binding to DNA. The platinum atom forms strong bonds, preferentially to adjacent guanine bases on the same DNA strand. This binding puts a kink in the DNA helix. It causes it to bend and partially unwind.
And that stops the cancer cells.
It severely interferes with DNA replication and repair processes. The damaged DNA eventually triggers cell death, and cancer cells, which divide rapidly, are often more susceptible. Interestingly, the trans isomer of cisplatin, where the chloride and ammonia ligands are arranged differently around the platinum, is almost completely inactive. Structure is everything.
Are there others?
Yes. Researchers have developed other platinum drugs, like carboplatin, to try and reduce side effects. And other metals like ruthenium are being explored for anti-cancer activity, too. There are also gold compounds, like sodium orothiamylate, which have been used for decades to treat rheumatoid arthritis, although their exact mechanism is still debated.
And finally, imaging. Seeing inside the body.
Metals are crucial there, too. For magnetic resonance imaging, MRI, complexes of gadolinium-3 are widely used as contrast agents. They alter the magnetic properties of nearby water molecules, making certain tissues show up more clearly. And in nuclear medicine, the artificial radioactive isotope technetium-99 99 millidC, is a workhorse. It emits gamma rays and has a convenient 6-hour half-life. By attaching it to different targeting molecules, doctors can make technetium complexes that accumulate in specific organs, heart, kidneys, bones, allowing them to image how those organs are functioning.
It really shows how understanding these fundamental inorganic principles allows for powerful applications.
Definitely. Human ingenuity, harnessing the properties of the elements, often inspired by how life already uses them.
What a journey. We've gone from tiny ion pumps to global photosynthesis to life-saving drugs. It really drives home how central these inorganic elements are to biology. They're the unsung heroes.
Absolutely. This deep dive, I hope, reveals this elegant universe of chemical precision operating inside every living thing. From the seemingly simple alkali metals setting up gradients to the transition metals performing complex redox chemistry and catalysis, NAM, K, lysise, charge, how they interact with water dictates their roles in gradients and osmosis. Ladroc, the main free divalent ion, key for ATP. A gentle Lewis acid catalyst and things like rubisco. K's structural in bones, but also that fast signaling due to rapid ligand exchange.
Then the transition metals.
M, unique redox capabilities, essential for water splitting and photosynthesis. Fe, the ultimate workhorse. Redox chemistry. O2 transport, catalysis, but needs careful handling due to toxicity. Co, specialized roles in B12 for methyl transfer and radical reactions. Nia, important in some microbial enzymes. Co, crucial once O2 appeared, electron transfer, oxygen reactions. Z, the perfect redox inactive Lewis acid for catalysis and structure, especially with DNA. Mo and W, masters of oxygen atom transfer from water.
And even things like silicon for plant structures.
Right, and then the non-biological metals we use in medicine, PT, AU, GD, TC, each chosen for a specific chemical property we can exploit.
It's an amazing toolkit, but this isn't just about understanding what is, right? Where does this knowledge lead?
Indeed. These insights are driving future directions. In medicine, understanding metal roles in neurodegenerative diseases or developing new sensors, like fluorescent probes for zinc in the brain. In energy, people are intensely studying enzymes like hydrogenases and that manganese cluster in photosynthesis, hoping to develop bio-inspired catalysts for clean fuel production, splitting water into hydrogen and oxygen efficiently using cheap, abundant metals.
Mimicking nature's best tricks.
Exactly. And in nanotechnology and material science, the way organisms control biomineralization offers blueprints for creating advanced materials with tailored properties. Self-healing materials, perhaps. Materials grown with nanoscale precision.
So the deep dive continues.
It really does.
Yeah.
And it raises, I think, a really important question for you, our listener. If life, through billions of years of evolution, can figure out how to use common elements under mild, wet conditions to create these incredibly functional catalysts, sensors, and structures, well, what complex problems could we solve in energy, medicine, materials? If we truly understand and learn how to replicate these ingenious natural designs, could we even create, say, new forms of biological computation based on ion flows, or truly self-repairing materials inspired by bone? The possibilities are vast.
A very thought-provoking challenge to mull over. Thank you so much for joining us on this deep dive into life's elemental secrets. Until next time, keep digging, keep questioning, and keep exploring.